All right, I think we'll make a start. This is sadly the last morning of chaos. I want to welcome you here, those of you who made it, given the wonderful evening we had last night at the castle. I'm sure there'll be some stragglers coming in, but nevertheless, I think we do have to get started. So without any further ado, our first speaker this morning is Pieter Medendorp, who will be talking to us about multisensory integration for stable perception and motor planning. Pieter.
Okay, thank you. Can you hear me in the back? Yeah. Okay, first I'd like to thank the organizers for inviting me to speak here. It's a real pleasure. I must say I've learned a lot over the last 2 days about things I did not know so much about, so thank you for all these fabulous talks. And also very nice to being in Rovereto, meeting people from the CMAC. It's also an institute that I admire quite a bit, so it's nice to be here and have the opportunity to speak here. So what I would like to do today is really talk about action. The conference is about concepts, objects, and actions. I thought about concepts. What do I know about concepts? Honestly, not too much. What do I know about objects? Well, to me, objects, they serve for action. And so what I would like to do is talk about this relationship between objects and actions and see what I can contribute to that notion with the research that we have been doing in our group. So the title of this presentation is Multimodal Integration for Stable Perception and Action. And so if you think about objects and we think about actions, I think there is a closed loop. So you need objects in order to drive actions, and you need actions in order to deal with representations of objects. Actions might disturb representations of objects. And so the question is, how does that process work? And it's really a closed loop in my view. There's no feed-forward pathway, there's also not a feedback pathway. It's both directions. And as I was saying, objects can be used in order to plan actions, and actions, they might perturb representations of objects. And the first component I call motor planning, and the second component I call perceptual stability. And I would like to talk about both. And so what I've set up a talk basically consisting of 4 parts or 2 parts, whatever you would like to, distinction that you would like to make. First about perceptual stability. And I would like to talk about 2 questions that we have tried answering over the last 2 years. The first one is on integrating memory and visual representations. And I would like to come up with the notion, and it also came up yesterday, about the causal brain. Trying to come up with causal inferences in order to make inference about representations that the brain constructs. And so that's the causal brain notion. The second then is related to that question is how does the brain integrate information that resides in different frames of reference, different coding formats. And I would like to come up with the notion of the optimal brain. So having discussed that, I would like to steer a little bit away from that, and I would like to show how can we use now objects in order to drive actions. And I would like to talk about integrating across effector systems. And then I would like to put forward the notion of the functional brain. And the fourth point then is about how we make these actions in different sensory contexts. Context can change. How would that change? And that's the last part of my talk. And there I would like to talk about the flexible brain. And I think these are all cognitive notions that I try to push a little bit with this type of research. And so the first question is about integrating memory and vision. The question of perceptual stability. And so now suppose that you have a number of objects in your visual world. For example, a house, a car, a tree, whatever you have. That projects on your retina. But we also know that we make frequent eye movements, these very rapid eye movements, saccades. We make them 3, 4 per second. And so that image of the world sweeps across your retina. And so the question is, and it's a very old question already in the literature, Helmholtz already asked the question, Descartes was thinking about it. So how do we keep representations of objects stable? How do we keep them stable across our eye movements? And people have thought about that a lot, and they came up with that notion of the brain makes predictions. And the idea is that if you make an eye movement, if you plan for a second, what you do is you take a copy of that command and you drive it to some internal representation, an internal model or a forward model. And the forward model takes that signal, but also takes information about the current scene. And it takes that model then, the output of the model is a kind of prediction of how the scene would look like after your eye movements. So that's what we call the predicted scene or the remapped scene. But at the same time, you make your eye movement. You drive your motor plant, you activate the visual system, and there's new reference visual information. And so what the brain now should do is it should make an evaluation of the predicted scene and the referent information. And evaluate, so what has now changed in the visual world? What has changed in relation to my objects? Where were they? So it probably should conclude that the house has remained stable, not so likely that that moves, and the car has moved. And that's evaluation process, that's what I would like to talk about. And many people have already discussed particular aspects of this model. For example, this pathway has already been outlined. People have thought about this. They've seen that neurons have anticipatory receptive fields that shift the receptive fields in anticipation of eye movements. But the question now here is, how does this evaluation process take place? What could the brain do? That's what I would like to answer. And so in order to address that question, we use a paradigm that comes from cognitive psychology. And that paradigm is called saccadic suppression of displacement. And so what does that mean? Well, it means that if you make an eye movement, if you make a saccade, you're not so sensitive to detecting changes in the visual world. You can do this in a paradigm, the saccadic suppression of displacement paradigm. And that goes as follows. And suppose you're looking at a display, 3 targets, a fixation target, a triangle, a circle, which I call the saccade target, and there's another target, non-target, a square object. And you ask subjects, well, fixate here. And now make a saccade. Make a saccade towards this target. And while making the saccade, all these objects on the scene, on the display, they disappear, as you can see. And one is slightly displaced. In this case, this non-target. Slightly displaced in the horizontal direction. And then you ask subjects, well, indicate the pre-saccadic location of that object. Where was it before the saccade? You can do this using a mouse click. And so basically there's 2 bits of information now. There's the pre-saccadic information, affected by memory representation, affected by the saccade. So make the output of the internal model. And there's this new post-saccadic visual feedback from this other pathway. And so the brain has to make an inference. What is this? Is this the same information? Is this the information from the same object or not? If so, it should perhaps integrate the information, because you get, as we know, as we saw yesterday also by Michael's talk, if you have 2 representations and these representations are both noisy, but the best thing you can do is integrate the 2, because you reduce the noise, you become more certain about your representation. And you can indicate where you thought the pre-saccadic location of the object was. And so that's the paradigm. Now the question is, how does the brain do this? How does the brain do this evaluation? What could the brain do? Well, you can think about 2 options. The first option is that the brain says, well, I have my pre-saccadic representation, I have my post-saccadic representation. Both can be described by probability distribution, so they are both noisy. And so this is the pre-saccadic one, and this is the post-saccadic one. What I should do is, well, I assume that's the same object I'm going to integrate. This is my integrated representation. And that means I put my pointer, my mouse pointer somewhere there. Brain could also evaluate, judge, well, this is a completely new object. If the object is shifted for, I don't know, a meter, you'd say, well, that's not the same object anymore. So it should ignore the post-saccadic visual feedback, only rely on the memory representation. And then you would give this output. And so this is a kind of either-or situation, a kind of selection mechanism. But if you think about this from an optimal control framework, this is not the best the brain should do. If the brain is a real optimal observer, it should infer the cause, if it's a stable object or an unstable object, but should put a weight to that idea. It should put a weight to that causal inference. And that's what we modeled. We can model this using causal inference modeling, and Michael already described it yesterday. And the basic idea here is, so you have the input to the brain, these 3 objects. They are both noisy representations indicated by these grayish ellipses. Then there's the post-saccadic input, the object that's shifted. And then, so basically the brain has to compare memory representations with a visual representation. And it should do this probabilistically. That's the causal inference idea. So it should compute a probability distribution. This is indicated here. The idea, the probability that there's a causal, that it's the same stable object that has been moved. And if it's a stable object, the brain should integrate. If it's an unstable object, we think it's not the same object, it should segregate. And so basically it should compute this equation. And I'm not going to describe this equation further, I'm only giving you the gist of the equation. And the idea is this. So you have your pre-saccadic representation, you have your post-saccadic representation, and the brain should make an inference, a causal inference. Are these signals coming from the same object? If so, then integrate. But it puts probability to that notion. And in this case, for example, it thinks 20% I'm certain it's the same stable object. So what it then should do, it should put 20% of weighting to the integration of the two representations, and it should put 80% on the segregation component. If the brain infers, well, I'm for 80% certain it's the same stable object, it should weight the integrated representation of 80% and 20% for the segregated representation. So that's causal inference. And basically people call this in the literature a mixture model. And we tested that mixture model. And we tested that in this paradigm. So we had horizontal displacements of the objects, vertical displacements of the object. We shifted all objects, the triangle, the circle, the non-target, different sizes of the displacement. And the post-saccadic visual feedback was presented for different amounts of time. Sometimes 50 milliseconds, sometimes 300 milliseconds, and sometimes a second. So that's the paradigm where we tested many subjects, lots of work. And so here I'm going to plot the localization error as a function of the displacement of the target. And first for that non-target, so for this target. So the target that was not in the saccade direction. So if subjects make no errors, the data should fall along this line, the x-axis. And if the subjects completely rely on the post-saccadic visual feedback, ignore the memory representation, data should fall along this line. And so this is the situation for that non-target, presented only for 50 milliseconds. And this is the data. So you see that there's a little bit of a non-linear pattern. You see that the data clusters a little bit along this diagonal line. But further away, bigger displacement, so the data comes back to that zero line. You see that better if you present the non-target for 300 milliseconds. You see pulling towards this post-saccadic visual feedback representation. But if the displacement is somewhat bigger, you bend back to making no errors, etc. This is for the 1000 milliseconds presentation. And so you can do this for the non-target, you can do this for the saccade target, you can also do it for the fixation target. And you get all these error patterns. And then you can see how well can the model account, this causal inference model account for these data. And, this is for the vertical displacements. To cut the story short, you can fit the model, and the model fits all these non-linearities in the data quite nicely. I think if you would put an R squared, it's like higher than 0.7. So that suggests that the brain really applies a kind of causal inference model to achieving this visual stability measurement. Then you can also test the model. So what can we now infer from the model? You fit the model, you can do some kind of reverse probabilistic analysis and say what kind of representations are probed by this model? Representations that you cannot access in isolation. You can only get to them by fitting the model and look at the model's components. And that's what we did. So we looked at a memory representation. So this is for the fixation target, the saccade target, and the non-target. The horizontal component and the vertical component. And you see here is the amount of variance, the sigma value. So it's about 1 degree. A bit more for the horizontal dimension than for the vertical dimension, which makes sense because the saccade is in the horizontal direction. So there's saccadic noise inherent in this representation. You can also look at the visual representation, and as you can see, if you present the visual target longer, the representation gets a little bit better. It's also, to us, it's quite sensible. And you can also look at how much do subjects now a priori assume that objects move or not move. You can also fit that value, and you get a value of about 50%. Subjects, on average, about 50% they assume objects will move or will not move. Yeah, so that model this causal inference model can really explain these data. So that brings me to that notion of the causal brain. The brain applies causal inference in order to achieve this perceptual stability of objects. First point. Now you can ask the question, so now how is this process being implemented at a more, so this is at a very computational level. If we now go one level down, how is it implemented? Can we learn something about that? And so if you look in the brain, and you look at how neurons encode information, you see that neurons use different frames of reference, various frames of reference. Some neurons code response fields in relative to the gaze center, an eye-centered reference frame or retinotopic reference frame. Other neurons use head-centered frames or body-centered frames, arm-centered frames. And many, many neurons use intermediate frames. They are neither eye-centered nor head-centered, something in between. So it's a whole mixture of reference frames that you can find in the brain. So it's very heterogeneous. And so now the question is, how could the brain use such reference frames in order to implement visual stability, this perceptual stability? And you could argue, if you have an eye-centered representation, you make a saccade, you move your eyes, you need to update eye-centered representations. But what happens to the other representations? Do they remain constant or not? Are they invariant to the eye movements? Well, we thought, well, let's test that. Let's test it by not only moving our eyes, but also move our body, so that at least gives the opportunity to change all reference frames. And the question is now, what happens? What happens at a kind of behavioral level, computational level? What can we argue? Okay, so now let's take, for the proof of principle that I'm going to describe, let's take two reference frames. And we move the body. And so let's take an eye-centered reference frame first. So a representation that is encoding space in an eye-centered reference frame. And I'll show you here. So what I'm showing you here is a kind of top view of a subject. This is the gaze direction. And these are two objects. An object a little bit away from the fixation point, and an object a little bit closer to the subject. Well, if the subject encodes representations, spatial representations of these objects in an eye-centered frame, you could argue this object 1, is to the right from my gaze center, and object 2 is to the left from my gaze center. If I position this subject at a different location, for example this one, you could now argue, well, object 1 is on the left from my gaze line, in the left in an eye-centered reference frame, and object 2 is to the right from my gaze line, to the right in an eye-centered reference frame. And so this object was initially, this position is to the right, and from this position it's on the left in my gaze frame. And so if you move your body from right to left, these coordinates of these two objects, they shift in opposite directions. And in visual perception, you call this parallax. So that's parallax information. So that's in an eye-centered reference frame. Now suppose that there is also neurons that encode space in a body-centered reference frame. Body-centered coordinates. Same two objects. Now we could argue, well, suppose that those neurons have as their anchor the body midline. So you could argue, well, both two targets, objects, are now on the left from the body midline. And if I move my body from this position, the two objects are on the right from my body midline. So if I shift my body from right to left, both representations shift, the coordinates shift in the same direction. Shifting. Okay, so two coordinate systems, and you see how these coordinates operate in these two reference frames. And so now the question is, if we keep this in mind, now what can we learn about perceptual stability? Again, I showed you this picture before. Well, suppose you look at a scene with objects in the scene. You have a tree, a house, the sun. So that's your retinal image. You have to process the retinal image using visual system, and you encode an eye-centered representation. Well, if you now move your body, you move your eyes, you need to correct eye-centered representations in order to keep them constant, correct them for the motion. You see a little bit this parallax effect. The house moves a little bit to the right, and the tree moves a little bit to the left. And then you can update your image. The brain could also do is say, well, no, I'm not going to use an eye-centered representation. I'm converting this eye-centered representation into a body-centered representation. I just take gaze position into account. So if I have eye-centered coordinates and I add my eye position, I get body-centered coordinates. Head-centered coordinates, body-centered coordinates. I can also correct that for my self-motion, get an updated body-centered representation. I subtract my gaze angle, and I get my updated retinal image. That's the alternative. And so the brain could use an eye-centered updating mechanism, it could use a body-centered updating mechanism. But again, that's not the smartest thing to do for the brain. If it would really try to get the most precise representation, it should do both. And so it should combine an updated representation in eye-centered coordinates, an updated representation in body-centered coordinates, integrate the two, and you get the most precise retinal image. And the question is, does the brain do that? And so the question now is, we know about updating in eye-centered coordinates, but is the brain also able to update in multiple reference frames? And so the question is this visual stability, this updating of representations, does it operate in parallel in multiple reference frames? Or does the brain just use this eye-centered representation and constructs the other reference frames based on that? So that's the question. And so we tested that in human subjects. So we put them on a so-called motion platform in a sinusoidal motion. And what we did is at the one turning point we present the target, and at the other turning point we present the probe. And we asked subjects, well, where was the probe relative to the target? And so we map out psychometrically the representation of the target. And so we had a number of targets surrounding a fixation point, so a grid of targets. And so we set up that model that I've just described in words. You can also set it up in math. So a target representation coded in an eye-centered frame, converted into a body frame. You include the self-motion in order to update these representations, and you combine them. And what we know from the literature is that self-motion perception is somewhat misestimated. There's a gain somewhat lower than 1, and of course there's noise. And the question is, how does that noise now penetrate into these representations? And that's the notion that we are going to use here. And the idea is that if we have this translational body motion, there is noise in the motion direction. But that noise plays out differently in a body-centered representation than in a gaze-centered representation. In the body-centered representation, the noise really operates along the motion direction of how you move your body. But because your gaze rotates while making the movement, that noise rotates in another dimension in the gaze frame. And that notion is what we're going to use, and this is really schematically. So if this is your body-centered representation, and this is your gaze-centered or eye-centered representation, if the brain is an optimal integrator, it integrates these two and it gets a very precise representation. Because it takes the best of the two worlds. It takes the best dimension, let's say in depth, and the best dimension in the azimuth direction. It gets the most precise representation. And that's the notion that we exploit. It's just exploiting geometry. And so that's what we did. So we have the model. We have these 9 targets in gray here, illustrated in the grid. And so we can look at the model. And the model makes predictions. Predictions if it's only updating the eye-centered maps, you get this in green. If it only updates body-centered representation, you get this in blue. If the brain is an optimal observer, an optimal combiner, you get this orange pattern. And so that's the prediction. So a big prediction is this overestimation. And that's what we can test. So we tested it using our paradigm. And this is the result. There's one subject in gray, how you localize these targets without any self-motion. This is how you remember these targets during self-motion. You see there's a little parallax effect. And so you can fit the model. This is the body-centered part, but not so well. This is the eye-centered part. Well, much better. But in combination, you really see that this optimal observer model really nicely matches the data. There's one subject, there's another subject. And again, now you can see here the body-centered part does a little bit better for this data than the gaze-centered, eye-centered part. But in combination, it's a very nice match to the data. And you can see this in all subjects. If you plot a measure for the quality of the fit, you see the lower the value, the better the model. You see the optimal integration model always has the lowest values. So what we argue is that this is the argument for the optimal brain. The brain optimally integrates spatial representations in multiple reference frames in order to implement that spatial stability. And that fits to that notion of the brain using multiple frames of reference. And of course, I can only show it here for a single or 2 reference frames. But the same argument holds up for all kinds of reference frames. But we cannot really test it geometrically. Well, as an aside, very briefly to get into a discussion, we also recently did the very same experiment, but now recording EEG while being on that motion platform. And we could show that there's areas in the parietal cortex that synchronize in relation to this updating behavior. And the activity really matches the updating gain that subjects show in this behavior. So there's some parietal synchronization that implements that visual stability process. And that fits to that notion of how the brain constructs spatial representations, right? So you have a scene, in which you subtract colors, edges, all of that, and you build locations, attentions, At the same time, you build object memories, periorhinal cortex, you heard about it. Of course, you also have the place cells in order to represent where you are in the world. But there's also all that information that is more in ecocentric coordinates, in which the parietal cortex plays a big role. Coding space in retinal coordinates, coding space in head coordinates, space coordinates. What we think is that these are the processes that we have probed with this paradigm. And we can even show that, working on a kind of neural net model that really mimics that parietal behavior. So that's coming out, hopefully. Okay, so that's this part of the loop. So we talked about the effects of actions on objects, perceptual stability. And what I've shown you is that the brain infers causality in the integration of memory and visual representations. And I've also shown you, I hope, that the brain is an optimal integrator. It optimally integrates information in multiple frames of reference. Okay, so now what about this part of the loop? So how do we use these objects in order to plan actions? What can we learn about that? Well, if we think about that problem, I think there's two very important components. If there's objects on the table, you have to select which object you want to act upon. You can only, let's say you can only act upon one object at a time. But you also have to know which effector you are going to use in order to make that act. Are you only looking at the object? Are you grasping the object? Are you kicking the object? So you also have to select your effector system. And so you have an object selection problem and you have an effector selection problem. And these two need to be integrated in order to plan action. And what we know from the literature is that the posterior parietal cortex plays an important role in that process. And then further up you have the further preparation, execution of the action. And the question now is, how does the posterior parietal cortex do this? And what's the underlying coding frame? Well, you can look in the monkey brain, and many people have looked at that. And if you look along the interparietal sulcus, what you will find is that the brain seems to encode representations for different effector systems in different locations. For example, in area AIP, the anterior end of the interparietal sulcus, you find representations that code for fingers or grasping movements. If you go more in the medial side of the interparietal sulcus, you find areas that seem to code more for reaching movements, hand movements, arm movements. And more on the lateral side, area LIP, seems to code more for eye movements. And that's what many people have shown. We actually have shown the same using MEG recently, or in recent years. You can also look at neuronal synchronization in the human brain, and you find similar areas, an area involved in making eye movements, a module of gamma band synchronization involved in reaching movements. So you see the same kind of organization. But the question now is this now evidence for a kind of somatotopic organization, or is this evidence for a more functional organization? Is this reflecting on what you have to do? Is it more action-based, or is it somatotopic? And what we thought is, well, we cannot address this question by only comparing eye movements and reaching movements. We need to include another effector system. You can argue functionally the eye has a different purpose than reaching. The eye inspects. For reaching, you manipulate objects. So let's add another effector system, and we added the foot. So we put subjects in an fMRI environment, and we asked them to either look at objects, reach for objects, or kick objects, point at objects with your foot. And then you find this result. So if you map out the representations for hand, foot, and eye, red, green, and blue. If you map that out, so this is the cortical surface, so this posterior parietal cortex. In the back, it's mostly bluish color, kind of eye dominance. And if you go more anteriorly, as you would expect based on textbooks, the foot more medially and the hand more laterally. And you can take a number of areas along the interparietal sulcus and you can see how the division of dominance is reflected. These little donuts. So you're really in the FP7. It's really on the junction of visual cortex and parietal cortex. It's really eye dominant, a little bit of hand and foot selectivity. And if you go more anteriorly, let's say this area AIPS, you see it's more dominant for hand movements. The question now is, what does this tell us? What does this tell us about representations of effector systems? Can we really tell something based on this evidence? And we thought, well, we need to look deeper. And that's what he did by using, and you all know this, perhaps even much better than we do, multivariate pattern analysis. That's also what we tried. And so, but we tried it a bit differently. And so, just to back up, so suppose you have an eye movement, and you make a reaching movement, and you look at the activation of a group of voxels, and the activation is equal. And what we can do is we can look at the patterns, as we know. And so this is the pattern for eye movements, and this is the pattern for reaching movements. And you can see, well, these are different patterns. So if you would put the information, the amount of information in a multidimensional scaling plot, this is where they are. So a clear distance between the eye and the hand movement pattern. So now we add the other effector system, the foot. As you can see, this is the foot, and if you look carefully, the foot looks a little bit like the hand. And if you compare the patterns are the same. However, if you look at the activation for the foot is different, much less. So now what would you conclude? What would you now conclude based on these observations? Would you say, well, this is a region that codes for eye movements? Would you say, well, this is a region that integrates hand and foot movements? Or would you say, well, this is a region that distinguishes eye from limb movements? So these are all the options that you could argue based on seeing this. So what we thought is what we need to do is we really need to combine the activation information with the similarity, the information. And that's what we did. And this is how we plot that. And so what we show you here is the distinct patterns on the brain. And so I showed to give you a sense for the coloring. And suppose, so we are now looking for distinct patterns. Suppose you have this situation, equal activation for all three effectors, but in a multidimensional scaling plot, clear distances between these three representations. Then I would color code that in white. If there's a clear distinction between the hand and the foot, but equal activation, I would color code that in yellow. If I would have clear distinction and similarity between hand and foot, but the activation for the foot is a little bit less than for the hand, I would color code it a little bit more orange. So that's kind of color code. And so now you can look at where do we find distinct patterns. And this is the result. And so again, as I showed you before, in the back of the brain, more bluish, eye movements, distinct patterns for eye movements. If you go more anteriorly, there should be some distinct patterns for hand and foot movements. And you can also pick up a number of areas along the interparietal sulcus, and you can plot them here in this plot. And you see the posterior interparietal sulcus is really blue, but the precuneus is really foot specific, and the anterior interparietal sulcus is really hand specific. So you can now find out where these areas are. You can also look for the common patterns. So which areas are commonly activated for groups of effectors. You can also set up a color scheme, and now it means if you have equal activation and no distinction in information, you color code in white. Or if you have equal activation, but only for hand and foot, and no distinction in the patterns, you would color code in yellow. So that means here's a common representation for hand and foot. That's what it means. And you can also map this color scheme out of the brain, on the brain, on the cortical surface, and this is what you find. A little bit of greenish color, a little bit of reddish color, not much blue. You can look at the various areas that we already indicated, and you can see where they are. And so in the area AIPS, you see a common representation for foot and eye. Etc. You can see where all these areas are. So if you now combine it in one plot, what you could argue is in the posterior interparietal sulcus, you only see eye-specific activation. In the medial interparietal sulcus, you see eye-specific activation and limb-specific activation. So common activation for hand and foot, etc. So you can follow how the brain combines effector systems or distinguishes effector systems across the parietal surface. And so this brings me to the notion that the brain is organized more functionally. Not so much somatotopically, at least the posterior parietal cortex. No, it's functional organization. The brain selects effector systems. Sometimes it combines them, sometimes it distinguishes them. And to me that makes that area suited as to function as a kind of selector. It selects an effector or effectors or classes of movements. And that brings me to that notion of the functional brain. Okay. How much time do I still have? Plenty? Okay, then the final point is how flexible is this coding? How can we now encode this information? How would the brain do that? Well, think about this problem. So if sometimes you have to act upon objects that you can see, sometimes you have to act upon objects that you hear, sometimes you have to act upon objects that you feel. You want to smash away a mosquito that's on your fingers. So how would the brain deal with those sensory conditions? What kind of coding formats, how flexible is the brain in dealing with these different types of sensory information? That's what we wanted to test. That's what we tested recently as follows. So we put subjects in a scanner again, and we either asked them to grasp visual objects or to grasp somatosensory objects. And these somatosensory objects were really locations on the opposite arm, as I show you here. So subjects were lying in the scanner. They saw 2 blocks. They were asked to, in this case, fixate somewhere on the left. And one of the blocks was acute, left block. The subject had to wait for a while, 3 to 6 seconds, and then they had to make the grasping movements, as shown here, with the 2 fingers. In this case, the left block, and sometimes the right block. And gaze was either on the left, sometimes it was in the middle, sometimes gaze was kept on the right. And so that's the visual condition. In the somatosensory condition, subjects had to grasp fingers or the wrist on the opposite hand. So it was in front of them, and they either had to grasp the fingers or the hand, or the wrist. And it was also acute. So you see the opposite hand here in front of them. And so this is grasping either of visual targets, visual objects, or somatosensory objects. And the question now is, how does the brain deal with these different sensory contexts? Okay, so what we have is the visual condition, and there's basically 4 different trial types. So what you could have is a situation in which your gaze is on the left, and you have to grasp the left block. You could have the situation in which your gaze is on the middle, and you have to grasp also the left block. You can have the situation in which gaze is center, and you have to grasp this block, or your gaze is on the right, and you have to grasp this. 4 different trial types. And the same for the somatosensory condition. And so now the question is, how are we going to analyze these data? What are we going to do? And we thought about this in the following way. Well, suppose we take 2 trial pairs. How can we now analyze them? Now, suppose we take these 2 trials. So that means in a gaze-centered reference frame, that means that these are 2 identical trials. In this case, gaze is on the center and the block is on the left. In this case, gaze is on the right and the block is on the right. But relative to gaze, it's the same trial type. So in this pair, these trials are the same. And the same holds for this. In this case, the block is on the right relative to gaze. If we look at trial pairs that are the same relative to the body, you have to take these guys. So that means gaze is here on the left. This block is on the left from the body, or gaze is on the right, and the block is also on the left from the body. Or this pair. And so what we now can do is we can run a classifier in order to distinguish these trial pairs. And so that means that in a body-centered area, that means these 2 trials in this pair are different. So you should be able to classify them in a body-centered area. These 2 pairs are, this pair is also different. The trials in this pair are also different. So you should be able to classify them if you're dealing with a body-centered area. If you're dealing with a body-centered area and you have these 2 trials, you should not be able to classify. They are similar. So you cannot make a distinction between them. So you cannot put a classification on them. Same for this one. However, for a gaze-centered area, it's the opposite. A gaze-centered area cannot distinguish these guys, but it should be able to distinguish these guys. Because these are different trials in gaze-centered corners. And so that's the basic idea. And so now we can also take it one step further. If we have a gaze-centered region and we are able to classify these trials, what we should be able to is to cross-classify these trials. So what do I mean by that? Well, take this situation. So in the gaze-centered region, you should be able to classify this. And so you have set up your classifier. If you now replace this trial with gaze on the center and the block on the left with the trial in which the gaze on the right and the block is on the right, it's a similar trial in gaze-centered corners, you should be able to classify it with the same classifier. That's illustrated here. So if you can classify this, you should be able to cross-classify this in a gaze-centered area. And the same argument holds for the body-centered area. And so what you can now do is you can take all the classifications and cross-classifications, sum them up, and you find evidence for the reference frame that is dominant in a particular area. And that's what we did. Yeah, that's what we did. Yeah, and the results are shown here. And so here I show you body-centered coding, and I show you eye-centered or gaze-centered coding in the warmer colors, and the body-centered coding in more cooler colors. And I show you this for the visual condition, in which you grasp visual blocks, and for the somatosensory condition, in which you grasp your hand, opposite side. And for the visual condition, you see more in the visual areas and also in parietal areas, you see this reddish coloring, dominance for gaze-centered coding. And if you go more in the frontal cortex, you see more bluish coloring. There's the representation already coded in body-centered coordinates. If you look in the somatosensory condition, you see much less gaze dominance, gaze-centered dominance. And you see much more body-centered dominance. That makes sense. It's grasping somatosensory targets coded more relative to the body. So that's what you can see. And so now what you can do is you can, again, indicate a number of regions of interest and see how well, what now kind of coding scheme do they now use. That's what we do here. And so I'm going to plot gaze-centered coding versus body-centered coding for a number of areas. For both the visual condition and the somatosensory condition. And here are the dorsal premotor and the ventral premotor areas. And as you can see, it's mostly body-centered. A little bit of gaze-centered, but mostly body-centered. The precuneus, also mostly body-centered. If you go to area Spock, the superior occipital sulcus, also at the posterior end of the interparietal sulcus, that's gaze-centered coding, where you present the target visually or somatosensory. It's gaze-centered. And if you look in area AIPS, it's interesting. You see this gaze-centered representation if you present the target visually, but you see a body-centered representation when you present the target somatosensory. And so that really shows a kind of flexible coding frame. It flips. If you present a visual target, the brain, the area says, well, I'm going to use a gaze-centered representation. If you present a body somatosensory target, the area says, well, I'm going to use a body-centered representation. So really a kind of flexible organization scheme. And that brings me to that idea of the flexible brain. So that AIPS is really a flexible hub. It just assumes the reference frame that is needed in order to convert sensory information into the body-centered areas as we see it. Okay, so these are my 4 points for today. So I think what I try to show you is that the brain infers causality in the integration of memory and visual representations in order to keep that perceptual stability of objects, the causal brain. I also try to argue that the brain is optimal. It optimally integrates across representations in multiple reference frames. I try to argue that the brain is functionally organized based on that effector type of selection mechanism. And I try to show that the brain is flexibly constructs representations in multiple reference frames. And that might depend on sensory demands, as I showed it, but it also might depend on your task at hand. And let me stop here. Let me also thank all the people, and mostly these people have done all the work. Jeroen Atsma, Femke May, Tjerk Guttelink, Mathieu Koppen, Julian Tramper, and Frank René. They have done all this work that I've presented here. Thank you.
Thank you, Pieter. I want to thank you and also for taking us through a lot of complex issues in a very accessible way, I thought. Absolutely. So, questions, please. Sharon.
So there was, in the last bit that you talked about, I don't think I quite got which conditions you used in the cross classification. So let me just ask you if this is what you did, yeah. And then I want to know about something I don't think you told us. So an approach we've taken to try and look at the informational content of areas with regard to distinctions that they care about is to train on a distinction in one set of stimuli and test on that distinction in a different, that same distinction but in a qualitatively different set of stimuli. So to look for a shape distinction in one color and test for the same shape distinction in a different color to show generalization. Yeah. And I think you didn't quite do that, right? You kept one of the conditions in your cross classification, or did you ever go to a completely different So in other words, did you train on the left-right gaze, left of body, and then test on left-right gaze, right of body, or you didn't quite do that?
That's what we did, but we did all the combinations that
You
are
did,
possible.
okay.
All.
Okay, so that was embedded. So in the data you showed us, sometimes you had the same condition and sometimes you had a completely different set.
Yeah.
So when you did the completely different set, did you go from vision to somatosensory or not?
Yeah, we also did that.
Okay.
Yeah.
So so I guess I was curious
that's not what we presented, right? We tested classification on, let's say, the visual target and see how that reflects in body center reps for somatosensory targets.
Right.
I did not present that here.
Okay.
Yeah.
So I'm curious to know what you found, and in particular with regard to the region where it switches. Was it IPS? I don't remember which one it was. The one where that was the basis for your flexibility, does it actually misclassify in those regions?
Yeah, it's a very good question. It's actually in the paper that we wrote. And I'm trying to dig in my memory.
So I should look it up.
I should have looked at that. But that question is exactly what we addressed, right? Can we, based on classification of visual targets, can we also decode or classify representations in somatosensory domains? And I think that basically it all worked quite nicely, but there were some exceptions, and I cannot remember where the exceptions were. Yeah, sorry. Yeah. The paper is out, yes. Yeah.
Questions?
So in the middle part, you focused on the parietal cortex mapping of different effector systems. Yeah. Did you look in other regions, especially the occipitotemporal cortex, where some papers have argued for effector representations in sort of cross-modal, or that are activated both by motor actions and perception of these effectors?
I know about that work. We have not been too explicit on looking at that, but it's not something that is standing out very obvious in our data. It's mostly more, let's say, parietal frontal organization that we see lighting up quite a bit in these experiments, whether that relates to the fact that it's really more a goal-directed act, other than also some of your work. If you look at tool areas or body areas, and more the extrastriate body areas or lateral occipital cortex. I'm not saying that we don't see anything there, but it's not something that really stands out in our data. And furthermore, I don't think we can decode reference frames in the way we do here. And the reference frame that we decode here are also more, let's say, ecocentric reference frames. And they seem to be a little bit more in dorsal areas, right?
I have a question. Well, I have a couple of questions. One is that often Efren's copy for eye movements is very clear. Efren's copy for body movements is a little more complex, obviously. And you had the vestibular organs up there as indicating a source of another source of information about how the body might have moved. So I just wondered, have you speculated about the kinds of information that's actually being used when you move your body? Often your body is moved passively, for example. If you were engaged in a tussle with somebody, then a lot of your movement would be quite independent of you, and presumably things like the movement of the scene across your retina would play an important role. And I wondered whether you'd thought about how this kind of stuff could be integrated into your optimization.
Yes, so in the end I think it's a kind of multisensory integration process, which is indeed Efren's copies, but also all kinds of sensory systems. If you do body motion, there's of course the vestibular systems that are important, but there's also the Efren's copy signal. We have done the very same experiment asking subjects to actively move, and we observed similar results. So suggesting, we have not compared the two explicitly, but you see similar types of patterns. Whether the pattern is a bit better, let's say in an active situation, because you have more, you have the sensory systems, but as well as the Efren's copies. And you could argue from a multisensory integration perspective, you would then get more precise representations. We have not been too explicit on looking at that, but at least you see similar patterns. The optic flow, of course, is also a very important component. If you move your body, the whole visual world shifts on your retina. The brain can use these cues in order to update representations. That's something that we are currently testing. So can optic flow signals help you in order to keep the world visually stable and remember where objects are? I think it will, also again from this optimal integration perspective. Where it breaks, or whether it breaks, in which situation it will break. That's something that we're currently exploring. And of course you can do that in a kind of setup in which you move subjects, you also move a visual scene, but you put some discrepancy between the two. You see when does the brain rely still on that optic flow information, and when does that break down? And you can put similar types of models, causal inference models, on addressing that question. But it's making the whole situation way more complex.
Yeah. I mean, clearly in the magnet, there's only so many things you can manipulate if you want to actually look how it's instantiated in the brain. And I wondered whether one could attack some of this by also looking at what you might talk about as a kind of temporal variable here, where clearly the vestibular system, the signal is going to fade pretty quickly, whereas maybe other pieces of information might linger. Have you explored the different temporal features of these sources of information?
The fact that the vestibular system degrades, if you have a sustained signal, after a while you don't sense it anymore because of the low pass nature of the system. Well, we have not explicitly looked at that. What we did do is in a type of different experiment in which you ask subjects to anticipate, perform particular cognitive tasks in sinusoidal motion, and sometimes you do it on the peak of the acceleration, and sometimes you do it at the peak of velocity. And you would argue peak acceleration is quite nicely detected by the vestibular system, whereas peak velocity is not detected by the vestibular system. And then indeed you see differences in cognitive behaviors. As if that signal, that vestibular signal, is only conveying that acceleration information. That's all I can say for now. But it's again a good question. But also that decay in the vestibular system is also there's still lots of velocity storage in the system. So before you really can tap into that question is a difficult one.
Right. Yeah, Jerry.
Thanks, Jerry. Just a quick question about the first part of your talk. So just wondering, so you focus on the brain making causal inference based on the translation, whether this object is the same as it was before the saccade. Can the framework be extended to include identity cues, which are obviously very powerful signals about, so qualitative aspect of the object was obviously a strong clue about whether this is the same token or not, right?
Well, I see no reason why that would not be the case. What is important for this causal inference notion is that you are able to frame that identity question in a kind of probabilistic framework, right? What is your probability distribution that reflects identity, and how would you encode it?
Yeah.
If just so, then you can generalize it to any situation. I don't see any problem with that.
Just that it's a very complex probability distribution, very many dimensions. I suppose that would be the main challenge. In practice.
I think the challenge is to really come up with a distribution that reflects identity in the most simple way, so you can also solve the equations.
Right. Okay.
Or simulate the equations. That's also a possibility.
Right. Thank you.
But I think I don't see any difference. It's just a
conceptually
kind of generic framework.
Yeah.
As also Michael showed yesterday, and other people have shown it for other systems. It's just a very generic type of Bayesian type of modeling that you can just apply, and it's more flexible than only assuming Bayesian integration always. Sometimes the brain says, I'm not going to integrate.
Right.
But how does the brain know? And that's that causal inference.
Right.
Yeah.
So I'll see if I can ask this under the haze of last night's wine. Back in the day, people used to talk about the reaching system and the eye movement system as sharing a common coordinate space, and that's how one integrated between the two. So what I want to know is, at one point early on, you were talking about the integration leading to you had some like an overshoot in I think it was the eye movements or the recall of where something was. I just can't remember now. But do you always find that do you find that where there is a kind of distortion due to the integration, that if you were to look at reaching movements, that they would distort in the same way as the eye movements due to the integrative underlying system?
I would assume so, but it's a good question. We had this paradigm, as we did it here, we cannot test it using reaching movements, because subjects are continuous in motion, and where do we have to reach? We have continuous at just the reaching arm. So therefore we did it psychometrically. You only probe it at an instant, and you say, well, what is it right, left? Yes, no questions, basically. If you would use only intervening motion from a standstill to a motion to a standstill, and you do then a reach, I think I would expect the same results. But I cannot really. I have to test it then. Yeah. But I would expect the same result. But whether that relates to the fact that there's a common reference frame for eye movements and reaching movements, of course, there's lots of literature that says that. But there's also literature that says it's a mixture of reference frame, so that encoding is a little bit more difficult than just assuming that it's a one single common reference frame that coordinates all the movement behaviors. Needs to be tested, but it's a good question. But for this paradigm, I would expect more or less the same result. Yeah.
There's a question over here.
Thank you, great talk. I have a question on the first part of the optimal integration. If I got it correct, in the condition in which the subject do the experiments, they have plenty of time for deciding where they see things. So do you think that under time pressure conditions, when the loop towards action and an object is also closed, you have the same opportunity and possibilities having always optimal integration possibilities? I mean, is it optimal integration mandatory, is always happening, or under time pressure there are more effector-located or effector-centered reference frames that can take over?
Yeah, that's a very good question. I would think that optimality by definition is always optimal, right? It only depends on the underlying signals.
It takes time.
Yeah, but if signals, that means that if it takes more time to get a more precise signal, then if you have less time, you have less precise signals, so your optimal integration might still work, but the outcome of that optimal integration might be less functional than you would like it to have, but it can still be optimal for the brain, given the circumstances which it has to operate in. So that's, and what I showed, for example, is that there is some time effect on optimal integration. If I present the post-saccadic stimulus a little bit longer, you've got more reliance on that post-saccadic stimulus. So time does something to the outcome of optimality. But whether that can refute optimality as a principle, that is a more philosophical question. Yes.
David?
Thanks, excellent talk. So at one point you had a slide where you showed all the different multiple reference frames having to do with perception, with action, and you also showed some nice data showing flexible changes in sort of the effector coding of the different maps. So given that at some point we make a single action and we have a single, presumably coherent spatial representation, how do you think the readout works out? So given that we have all of these different maps, how do we converge on a single action, or we converge on a single unified perceptive space that includes all these different maps?
Yeah, how do you do that?
That's the $1 million question, $1 million question. Well, there might be different ways for the brain in order to align, for example, representations and come up with a representation that could serve your actions or could serve your perception. Some people claim, for example, that neurons start to synchronize. Neurons that have importance for a particular task, they start to synchronize. And that's also what we see. If we, for example, as subjects to reach, we see that particular groups of neurons start to synchronize in particular frequency bands, and that there's that even frequency bands of neurons that code in different frames of reference start to synchronize at the same frequency. So that could be a mechanism for aligning up representations. In the end, whether that is the mechanism, you can always ask, well, how does that synchronization come about, right? So that you got one level deeper. I'm not sure if if there's already that there's neuroscience work that really shows how the problem is solved in the brain. I think synchronization and face coding, these kind of mechanisms might be important for that behavior. But in the end, you can also argue whether there's just an epiphenomenon from what we see in the brain, whether it's really the mechanism for the brain. And there's also still lots of debate about that question. I find an interesting question. If you build a neural network model with basis functions, you can see you can mimic all kinds of behaviors that you would like to serve with the same group of neurons. Neurons can encode so many patterns. You only have to select the right pattern in order to get your behavior going. And in a neural network, you can encode that. In the end, it brings you to where's the soul, right? When do we switch on? And when do I decide to make a reach or to make a saccade? These are difficult questions. Yeah. Is that answering? Do you have thoughts yourselves?
Yeah.
I'm not going to solve it.
Yeah. Richard Gregory always used to say when he had a difficult question, that's a great question. What do you think? So
that's what I tried.
I wondered what happens with skill. For example, the right hand versus the left hand in many occasions, like particularly manipulating objects, right-handers more often used. Do you see any differences in your fMRI studies in the way in which the left and right hand, for example, are represented? With respect to reliance on particular frames of reference, do you rely in an unskilled action more on, say, gaze-centered coordinates than you do on limb-centered coordinates?
Yeah. Well, so far I have to admit that the tests that we do, they are very, very simple. They are,
right.
The objects are very simple. They are just points. The actions are very simple. They are just goal-directed acts. Either make an eye movement to a single point or make a reach movement to a single point. Maybe grasp it. That's the most complex situation that we have. Where this gaze-centered reliance is, and that's a good question. Last week I was at the Neural Control of Movement meeting. There was a whole session on what's the importance for gaze-centered coordinates. People see it in monkeys. For example, if you train if you study monkeys in these types of paradigms, but the monkeys need to be trained, and you see the gaze-centered representations. If you just test monkeys without this gaze-centered training, you won't see much gaze-centered coordinates. So it's a very, again, this leads to this notion of flexibility. And that can also be a criticism of the work that I've presented. Is this something that we see because we have these paradigms, they are so much a kind of laboratory paradigm, it's not very ecological. So what we see is really something that comes out because we set up the experiments as such. That's a good question. How the brain would, the representations that the brain would rely on, let's say in more complex situations, and whether gaze-centered coordinating is equally important in those situations. I think that's another important question, and the solution to that question is also a difficult one, because the question is, how are you going to test that?
Yeah.
I believe that what we see is, of course, in some ways it's trained behavior. We humans, we manipulate objects all the time, we look at objects all the time. Apparently, that there's gaze-centered reliance. I cannot ignore that we see that in our paradigms and that others see it in their paradigms. But whether you can go without, I would think so, honestly.
Right.
Ask Mel what he thinks.
Yeah, Mel, what do you think?
Yeah. That's a question for a future day, I think.
Oh, Alex, what do you think? Maybe you have thought. What do you think?
Any more questions? I foolishly came up here without the program. I do believe that we reconvene at 10:45. All right, so posters and no posters. Coffee. Back here at 10:45.